Hey everyone, it's Mr. Sinti, and today we're going to talk about the making of protein, and we're going to talk about how DNA codes for protein. It's the recipe for protein. The information to make a protein is located on DNA, and specifically in the sequence of nitrogen bases in DNA. And that, as you may recall, is called the gene, a segment of DNA that codes for a polypeptide. But I want to talk to you today about an intermediate molecule because DNA, though it codes for protein, doesn't actually make protein directly. It provides the instructions for making a protein, but it doesn't actually get the job done. And so this is my question. I want to start off with a little bit of a question for you. In other words, how come DNA doesn't make a protein? I mean, is it lazy? Does it not want to contribute to this? Why does it have someone else doing its work for it? Well, the answer to it is that it doesn't do it directly, but it copies itself into a very similar molecule called ribonucleic acid, or RNA, and that's what this conversation is today. RNA is going to be the recipe for making protein, but again, why? Why is it that RNA is directly involved in making a protein? You can see DNA, the double helix here. Here's a protein, which is a chain of amino acids all coiled up in this interesting shape, and these helix and sheets like this. So this is a three-dimensional structure right here. Why does RNA have to do it? Why not DNA? DNA controls the recipe for making protein. Well, to me, it's kind of like this. I mean, you could have a recipe book, or you could be the chef at a restaurant and be able to make every single recipe in this book. But don't you think it's wasteful every night to be making every single recipe? It would be more efficient to only make the recipes that someone ordered. Or in other words, if you were a cell, why would you make every single protein that you possibly can? Like, for example, your skin cell doesn't want to make every single protein imaginable. Because I think ultimately it isn't that DNA is lazy, is that it conserves energy. The cell conserves energy by only producing the proteins that it needs. And so therefore, RNA is sort of like a photocopy of a recipe that is needed at the moment, and then that travels to the ribosome to make proteins. And so therefore, RNA is something that can be an intermediate, and it's also something that's temporary. It's sort of like today's news. It comes out, it gets the jobs done, and then you recycle the RNA. And so look, that's what our conversation is going to be about today. So hopefully that's intriguing you. And so let's go up here and get a larger view. So as we said, DNA is the instructions for making a protein. We won't talk about it in this particular lesson, but the sequence of nitrogen bases codes the genetic code or the recipe for making a protein. But this is what I want to talk to you about today, which is RNA, or ribonucleic acid. It specifically controls protein synthesis. And so DNA is really huge. And again, we mentioned it has the recipe for every molecule, every protein. So it goes off really far, and I'll just do this. It goes off really far in both directions. And so if this is an area right here, let's go highlighter. If this is the recipe right here for one particular protein, what's clever about it is that DNA can open up and it can make this molecule called RNA. Now RNA is very similar, you might imagine, to DNA. It's ribonucleic acid, or RNA. And so we're going to look at the structure of RNA today. And we'll talk about how it's transcribed from DNA in the next lesson. But here's RNA. You might be able to tell just by looking at it that it's often single-stranded, and it's very, very similar to DNA in a sense that it's a polymer. So let's talk about that. And so DNA controls protein synthesis by making RNA, and then RNA goes out and gets the job done on the ribosome. So let's look at an example of what we're talking about here. Like, for example, what job needs to be done? Well, proteins are responsible for getting the things done in the cell and getting things done in the whole body. We're constructed of protein, but DNA is the recipe for this. And so what does this all mean? This means that, for example, in a cell in your intestine, DNA contains the recipe for making proteins. Here's an example of that. A protein that you'd be needing if you were to drink milk is the enzyme or protein called lactase. Here's milk sugar. Milk sugar. You might be familiar with this example. Milk sugar is called lactose, and so the cell needs to make a protein called lactase. And lactase needs to come along and digest milk. And so, all right, where is lactase made? It's made on ribosomes. That's correct. But who controls this? And it's like, well, amino acids need to assemble to do this. And so ultimately, RNA is going to direct the amino acids in the correct sequence in order to make the protein. And so DNA is going to make the RNA, and the RNA is going to make the protein. So there's our little central dogma, if you will. RNA is going to control that process. And so, why do we need proteins? We need proteins for many things. Like, for example, if you didn't produce lactase, then that undigested sugar would pass through your small intestine into your large intestine where the E. coli live. The E. coli would be happy to break down lactose that hasn't been broken down. And when it breaks it down, it produces a lot of carbon dioxide. And you know what that means? That there's going to be some southerly wind coming out this hole down here. And so here's another example of this. Like, say the substrate was lactose, the enzyme is lactase, and so the enzyme breaks the substrate down into the products. So proteins get the job done, but RNA is what is the recipe for making a protein. And it's like, let's just talk about that for a second. And so remember, a protein is a chain of amino acids, like this. It's a chain of amino acids, a protein. It's more than just these few. But the thing is, in order to have the correct shape, you remember how enzyme works? It has to have the exact same shape as a substrate for it to be able to speed up a chemical reaction. So in order to have the right shape, you need the correct amino acid sequence. And so RNA is going to provide that recipe. And it's like, well, how does it do that? Well, it's in the nitrogen base sequence. It's the nitrogen base sequence which is going to tell the amino acid sequence. And so this is something we'll look at later, although I'm sort of foreshadowing it. What does RNA look like? This is my real question today. What does it really look like? And so, again, DNA is stored in the nucleus, and it's too big to get out. It does get out during mitosis, as you know. But it lets RNA do the work for it. So small pieces of RNA are copied in the nucleus or transcribed, and then that RNA travels out of the nuclear pore and goes to a ribosome and makes a polypeptide or a protein. In this case, it could be an enzyme. That's what we were talking about a moment ago. So what does RNA look like? Well, first off, it's a polynucleotide. And so let's look at that, a nucleotide. So just like DNA, a nucleotide is composed of a phosphate connected to a sugar connected to a nitrogen base. So let's go nitrogen base. And there is a nucleotide. So if it's a polynucleotide, you're going to have many of these connected together. Like, well, is this exactly the same as DNA? It's very similar. So we're going to focus in on some of the differences between an RNA nucleotide, which is the monomer, and contrast it with that of a DNA nucleotide. So again, as I mentioned, a nucleotide is a pentose sugar, a 5-carbon sugar, a nitrogenous base, and a phosphate group. And so let's take a look at this diagram. It's very helpful. As you can see here, here's your typical nucleotide. It's a phosphate over here, negatively charged. Here's the 5-carbon pentose sugar, and there's the nitrogenous base. So what's the difference in the sugars between DNA and RNA? The difference in the sugar is that in RNA, there is a OH group, or an oxygen-containing hydroxyl group, coming off of the second carbon. So deoxyribose is found in DNA, and that's why it's called deoxy, because it's lacking the oxygen in the hydroxyl functional group. So it's without the oxygen. I'll just be silly about it. So I'll do this. It looks a lot like ribose. See this? Ribose. deoxyribose. So if I put that, then it would be ribose. And so that's one of the differences, is that the sugar is different. Another difference is that one of the nitrogen bases is not present in RNA. Thymine is only present in DNA, so T is only found in DNA, and what replaces it is another pyrimidine called uracil. And so uracil is, if you look closely, compare it to thymine, it's pretty similar. The only difference is a CH3 coming off of that carbon right on the side there. And so uracil is the difference. So in terms of the 4 nucleotides, we would go phosphate, ribose, sugar, and then uracil. Phosphate, ribose, sugar, cytosine. Phosphate, ribose, adenine, and phosphate, ribose, guanine. So these are the 4 nucleotides of RNA. Pretty similar, different sugar, and uracil is the difference. And so, again, here's the emphasis of RNA. It's the sugar ribose, which is the OH coming off of here. And so that's the big deal between RNA and DNA, ribose, and that's what the R stands for in the word RNA. And then uracil is slightly different than thymine. It's still a single ring called a pyrimidine, and you can notice the difference here. I can make it thymine. Like here's uracil, but if I put a CH3 right there, then it would no longer be uracil, it would be thymine. But notice this side is similar. So as we're going to see when we talk more in detail about how RNA is made, it can still hydrogen bond. Whoops, over here. Oops, didn't mean to do that. Let's see here. Hopefully I can get that back. Sorry for this little delay here. I'm not sure why that's doing what it's doing. Okay, ooh. Okay, I'll just be thankful. The desktop scribble has quit. Sorry for these technical difficulties. We're okay. Okay, let's go back here. Sorry about that. Okay. Okay, let's go this way. My point was a simple one, in that uracil can still hydrogen bond with adenine, just like thymine can bond with adenine. And the way it can bond is that if this is adenine over here, it can hydrogen bond over here and hydrogen bond over here, because this side of uracil is similar. That was the point that I was trying to make before things got a little crazy there. So in conclusion with the slides is that DNA is a double polynucleotide. It's double-stranded, and so this is just one side of it. And so you can see here's the deoxyribose sugar in blue, and then it has thymine over here. And RNA happens to be single-stranded most of the time. And it's much shorter, obviously, than DNA, because it's just a copy of a small area. And you can see uracil over in this direction. And so let's go down here. And one of the things that I wanted to say in conclusion about RNA is that being that the fact that it's single-stranded gives it opportunity to sort of move around a little bit and be more creative in terms of its three-dimensional shapes. And so that's going to afford it some interesting possibilities, because in the next lesson we're going to talk about there's a couple of different functions of RNA. One of them is to carry the genetic code. Another one is to transfer amino acids to a ribosome. And believe it or not, one of the RNAs actually comprises the ribosome itself. So you can see how it can fold around. It can even form hydrogen bonds with itself, forming like really interesting looking structures like that. And so I hope you enjoyed this conversation about ribonucleic acid. Thanks for watching.